Good afternoon. I just want to start by thanking the organizing committee for inviting me to this meeting. I've known about this meeting for a long time, but for various reasons have never had the chance to attend. So I'm really grateful to have the chance to be here. So what I'm going to do today is share with you kind of the latest results of a project that we've been working on for a little over 10 years. My outline here, we'll talk a little bit about just sort of general concepts in lactation genomics and comparisons among humans, mice, and cows. I'll then go on to present data that we've analyzed to produce some candidate lactation QTLs in the mouse genome, and talk a little bit about this broader resource that we've been working on that we now refer to as the mouse lactation phenome. As a result of these efforts, we, a few years ago, a year and a half or so, decided we needed to go back and do whole genome sequencing on a number of the strains that were a part of this cohort. And then the last two parts, just some efforts we've had in progress to integrate mammary transcriptome in selected strains with our whole genome data, and potential opportunities to do more comparative work to try and really get a good way to identify the functional candidates, because that's where it's at. So, all right. So I love this slide as the first part of my talk because it really illustrates the large amount of phenotypic variation that you have in inbred mouse strains. And when I got interested in this, it was still very early in terms of the genome, but even at that time, there was a lot of resources available in terms of public databases on genetic variation in the mouse. So you could go to these and they were available at the Jackson Laboratory. There was a site there called the Mouse Phenome Database, and you could download them and use them. And the thing that really excited me was this idea that you could basically order these mice in groups. As inbreds, they're generally very homogeneous, and then phenotype them and have the genotype data set readily available to then start to do GWAS with. My sort of preliminary comparative data, since I've been working in the mouse but at a human research center, I've always sort of tried to, at least as much as possible, think about these comparisons in terms of the human and the cow. And of course, there's my pointer deciding to do some strange stuff all of a sudden, but there it is. Anyway, it's obvious that they have general differences in just overall strategy, but also, I don't know why the heck my pointer is moving all over the place. Might have to stop that. Anyway, that's really weird. That's unbelievable. I don't know what the heck's going on with it. All right, ignore the red dot for now. So you have differences in lactation strategy. And of course, if you look in the lower right-hand part of the slide, you can see the dramatic differences in the composition of their milk. One of the things that's often fascinated me is this idea that there are dramatic differences in the output of their energy relative to the size of the offspring that they're trying to rear. So the graph is a modification of some early data from Olaf et al. That compares energy output as a function of the size of their offspring. And over the years, I've been able to add data from other published studies so that we can enhance the number of species. But the main thing to note is that at the very top of the graph in blue, you can see the mouse, the top right corner of the graph. And then if you get down to the bottom lower corner, the human is in gray. A few years back, I managed to get some data on dairy cows. So the cyan and the green symbols represent Holstein cows from 1976 and 2016, where I managed to get kind of an average from DHIA records. So you can see that there are definitely outliers from the curve, but they're still relatively low relative to the mouse. So these are things to kind of keep in mind when thinking about these comparisons. This is sort of the state of the GWAS situation when you compare cows and humans and mice. This slide probably needs a little updating. I wonder if maybe when I was rehearsing this, my pointer got recorded. Anyway, you can see it's pointing to the cow table there, and there's about 5,000 to essentially tens of thousands of loci in the cow, and there's been a few clear genes that have been identified. But then also, if you compare that to the mouse, once we got started, there really were a relatively low number of loci, and we've added a few to that. With respect to the human, it's still a pretty bleak situation. There's now some work that I'm aware of being done at the University of Idaho, Shelley McGuire's group, where they've started to try and map QTL in breastfeeding women linked to different milk composition aspects. So hopefully that'll allow us to advance that in terms of comparing these three species. I want to go through this relatively quickly, but in thinking about comparative genomics, this is data from the Ensemble resource. This just compares the size of the genomes and how many genes there are. They're all roughly the same in a lot of these things. If you look at how much conservation there is among the three species, they all have about 20,000 orthologs that you can sort of call genes that might be analogous in the three groups. And so that gives you something to work with. Where they're very different is in the known genetic variation. So if you look at the bottom, you have short variants and structural variants. And in the human, there's only there's on the order of almost 700 million that have been identified. In the cow and the mouse, they're more like 100 million of those each. And that's partly due to the fact that sequencing has become so cheap, and there are a lot of groups around the world that are trying to sequence hundreds of thousands, if not millions of individuals. So this paper was really what first got me interested in this aspect of research, and it was a study published in 28 where they really resequenced 8 inbred mouse strains, and they were able to create an initial research source of about 8 million SNPs. And then what they did was to use that data to do kind of a phylogenetic mapping to show the origins of the laboratory mouse that we commonly use, which is shown over here in the right, this multicolored one. But essentially, you have shown here 4 subspecies of mouse, and we know of others as well, at least one more. And these originated in various places, Eastern and Western Europe, Asia, and at some point, a couple of 100 years ago, people got interested in these esoteric things like coat color and curliness of the hair and things like that, and that's how they started breeding them, and eventually gave rise to what we have today. So this is what we're dealing with in terms of the genome. You can also do other things, and this is a little bit of an old slide, but this shows the relationships among the different known inbred strains that you can get from places like the Jackson Laboratory. And we use this to identify our initial study. You can see they're broken into about 7 groups, wild-derived and CC's little castles, C57 related, Japanese and New Zealand, Swiss and bag albino. And for us, we wanted initially to at least get strains that were representative of those different groups. So all of the ones that are shown in cyan, where the strains were, we were able to get a set of founders from the Jackson labs and then produce study animals from those. So the work was started, and this was in 2011 where we originally did this collection, and it was done with the help of Peter Williamson, who's at the University of Sydney. He had a student, Jerry Way, who came over and worked as a postdoc in the lab for a while. And in fact, we still have a lot of samples from that original collection that he helped get. But what we had were 31 inbred strains, and we studied them over the first 10 days of lactation. So the 10-day period, they were all second parity. And the sort of timeline is shown on the bottom. The dams would give birth on day 1. We would cross-foster them to a weight normalized litter of CD1 pups that was also day 1. And we'd look at some traits like maternal behavior and body composition in the mom. And then on day 5, we'd look at aspects of how long the mom spent on the nest during the light phase or the dark phase. On day 8, we did a weigh-suckle weigh to try and get a separate indication of milk yield. 9 was, again, another body composition measurement day. So we could not only get body composition of the mom, but how her body composition changed during that 8-day early lactation period. And we'd collect milk, and then day 10 was the last day where we had an opportunity to measure an indicator of milk ejection, and then we'd collect tissues. So just a little brief discussion of GWAS, Genome-Wide Association. This is a kind of a part of a tutorial that was published a few years ago. And it's essentially based on the idea that you study some trait in a group of what you assume are unrelated individuals, although at some level back in time, we know that there are ancestors that tie them together. And then depending on your trait, you stratify your individuals by what that trait is, either a case or a control, or high or low body weight or whatever. And the groupings of those hopefully are going to then associate with some ancestral mutation. That's your SNP that could be potentially causing part of the trait that you're interested in. For us, when we first got into this, there was a fairly low amount of data available, and this was what we were able to download from the Jackson Laboratory, about 600,000 SNPs. After filtering them for quality, we ended up with about 300,000, and then from that, we could use this package in R called EMMA, which allowed us to use mixed modeling to then do the association analysis. So this is one of the first traits that we were really interested in, and this is litter gain, cross-foster litter gain among the mice. On the left-hand side, you see a heat map, and what this heat map shows is the daily change in gain of the litter. The x-axis is days postpartum, and the y-axis are the strains, and they're clustered by their ability to support the growth of the litter. So you can see at the top where there's red, those are the high lactation strains. And at the bottom where you see more blue, those are the low lactation strains. And there's a few that jump out in particular, QSI5, which is that dark red band near the top, and BTBR. Those are very capable of supporting high litter growth. And some of them, in fact, give birth to very large litters. And then at the bottom, you have these strains like MAMI and PLJ, and AJ. And in fact, in the graph on the right, what that shows again is the daily increase in litter weight as a function of time. And they're normalized to their initial litter weight. So on day 1, they start out all at 0, and you can see the circles in the different shades of gray are our high lactation strains, and the colored diamonds are the low lactation strains. And even by about 24 hours, there's a pretty dramatic difference in the ability to support the growth of these litters. So in addition to milk composition traits and body composition, we also measured some maternal behavior traits. And I like showing these because of the fact that, let's see if I can actually get this to work. It's not. I'm going to actually escape out of here for a minute because I do want to see if I can get this video to work. It's a lot of effort, but there it is. So I'm going to turn that off, and it is off. Anyway, 2 traits. This stretch reflex trait shown in the video in the middle is at L10 mom suckling 10 pups. When you see that huge stretching response, that mom has just had a milk ejection episode. So it's an indicator of basically oxytocin secretion. And in a normal lactating dam, once, if they've been separated from their pups for a while and you put them back, after a few minutes, you'll see these every 7 to 10 or so minutes. And so that was one of the things we were able to measure. A second one on the other side is Pup Retrieval, which is also one that's commonly used. This was something we measured in L1 dams. And if you pull the litter away from the nest and separate mom and then put them back on the other side of the cage, they'll very rapidly retrieve them within less than a minute. Both of these traits, I show that diagram of the pituitary and the baby suckling in this axis because both of these traits are dependent on oxytocin. So just something that I enjoy about one of the things we measured in this cohort. Okay, so what this heat map then shows is the collection of all of the maternal traits that we've measured in this study. On the y-axis are our proxies for milk production. There's litter gain during the first day, an average gain over 8 days, a weigh-suckle weigh measurement, how much milk we could recover, and then lastly was just how many pups survived that first 8-day period, because in some of the low lactation strains, pup viability could be an issue. So the heat map is actually Pearson's correlation, and you can see the key there. The bright gold is positive and the bright green is negative. And then the traits are colored by sort of these basic classes, say milk yield and milk composition, maternal behavior, maternal body composition, fertility, mammary metabolism, mammary development, and maternal neoendocrine function. And so when you look at these, you can see that basically all of the milk production related traits cluster and are the most highly related to each other. And that's not surprising. They should be measuring the same thing. But then there are a few other traits. First of all, that's one of our primary ones that are sort of more related to those. And so that's what the cyan arrows show. And one of the most closely related ones was maternal food intake. So you can see right there on the sort of a little right to center, there's dam food intake, and that's about a 30%, 46% correlation with litter gain. And that was the strongest one. Also, maternal weight was very strongly correlated. And then some other things, for example, mammary total DNA, mammary gland wet weight were positively correlated with litter gain. Interestingly enough, we have been able to measure all of the fatty acids in the milk samples that we collected, and this is something we did with the help of Kevin Havertine and a grad student of his who's here, Cesar Matamoros. He has a poster on this afterwards, but we looked in this case just at sort of the summary fatty acid traits in the milk, and one of the things that came up is on the right side of the graph. Well, if you look at milk total de novo synthesized fatty acids, there's a positive correlation between that and litter gain. And interestingly enough, but not probably surprising, on the left side, there's the sort of amount of milk preformed fatty acids, and those are negatively correlated with litter gain. Okay, so just to kind of summarize then where we've come with this, this is an ideogram of the mouse 20 chromosomes, actually 19 in the two sex chromosomes, and you can see the symbols along each of these chromosomes are indications of where we've mapped a locus. In some cases, we've been able to find a similar locus in the cow. More recently, the work we did with the milk mineral composition, there was one example of that we were able to find. And then the papers coming out of this have been really slow, unfortunately, but we have got some. And I'm going to spend a little bit of time talking about what we found with the milk mineral analysis that we published most recently. So one of the interesting things that we found right off the bat when we looked at milk iron, this was among a group of 9 minerals that we measured in the milk, was that there was this locus on chromosome 1 shown here. We referred to it as MMCQ9. It's actually mouse milk mineral composition QTL9. But what you see here are the strains that were measured, and the genotype of the SNP is shown on the bottom. So the 0 is the wild type allele of the SNP, and the 1 is the alternate allele. And you can see most of the strains that are in the wild type group have an average iron concentration in the milk of about 14 or so micrograms per mil, but there are 2 strains in the alternate group that have about 40% more iron in their milk. So this was really something that piqued our interest, and we went on to focus on this region in a little more detail. So this is a busy graph, but what it shows is a way that you can try and identify the other variants that are linked to your variant in the chromosome or in the region, and identify and sort of look at those as potential causal loci. I need to just go back and state, and I'm sure some of you or many of you are already aware, just because we have an association doesn't really mean that we have a functional link. And so this is where the work becomes a lot more challenging and slows down. So what this graph shows is each symbol is a SNP that's an association. The left y-axis shows the log of the p-value, the negative log of the p-value. So the higher it is, the more significant it is. Along the x is the location on the chromosome. And then on the y, what we have is stat binding regions. And so that light blue region of peaks, those are areas where ChIP-seq data from Lothar Henninghausen's lab has shown that STAT5 is bound in that region. So that was something that we sort of used to try and look at something that was more specifically related to the lactating mammary gland. The other thing that I'll point out is, so the SNPs are colored by their linkage to the lead SNP, which is the large blue diamond. So the red ones are highly linked, and the statistic here is R squared, derived from this LD statistic. So those are above an 80% R squared, which means they're highly linked to the blue. And then as you go down in color, with the dark blue being essentially less than 0.2 of an R square. So you can see there's a region there, and then you also have a number of other variants in the region that could be causing your effect or not. So those are candidates to investigate. So the way we then go about doing this, and this is work that Monique Wrinkles does to contribute to the effort. She has tracks on the UC Santa Cruz genome browser and in other places where she has information on how different parts of the chromosome have epigenetic modifications, histone modifications, or modifications like DNase 1 hypersensitivity peaks that indicate a gene is being transcribed. And then lastly, data from ChIP-seq to indicate if there's specific transcription factors binding. And so the diagram on the left kind of indicates how we go about doing this. You have, let's say, your lead SNP in red shown there, and then at the bottom, you can see the linkage disequilibrium statistics. And the idea is that you have SNPs nearby that may actually be the causal SNP. So for example, SNP1 is the lead, but it's likely that SNP6, the blue one on the other side, may be the causal one because it happens to reside in a transcription factor binding site and maybe change the sequence. And it's also underlying peaks of DNase hypersensitivity and transcription factor binding. So what we did then was to take all of the high LD SNPs in a particular region. In this case, the table shows how we went through this, but we started out with about 4,600 SNPs, and then we can whittle those down to smaller numbers as we overlap with different features. So DRE is a distal regulatory element, and we had 624 of those. And then if you cluster binding sites together in these, those are called core elements, and they're defined as having DNase 1 hypersensitivity as well as histone modifications that indicate open chromatin. And then further down, there are tools like this Homer tool that define the transcription factor binding sites based on historical data of what they're supposed to look like. So you can see the list gradually growing smaller. When we get down to STAT5 binding regions, there's 14. And then if we use Homer and we actually find a STAT5 binding site that's changed as an effect of the actual mutation, then that's where we ended up. So we ended up down with 6 potential candidates as a result of this. This is just another look at that, except I've decided to remove the genes. The other thing you do, of course, when you have this huge list of genes is you go back and you say, okay, based on what I know about the biology, what are the most likely candidates? And in this case, we decided to focus on 4 transporter genes that were in the region, these SLIC genes. So you have SLC9A4 and 9A2, which are kind of in the middle, almost underneath the peak. And then towards the right side of the graph, SLC40A1 is also known as ferroportin. And so that's important for transport of iron into vesicles and maybe playing a role with secretion of iron into the milk. And the last one is a zinc transporter, 39A10. So these were potentially good candidate genes to follow up on as well. Now, as a result of the analysis that we did in the previous slide, we decided to focus on this one particular SNP that was right underneath our region of interest. And again, Monique did much of this work, but what she did was to look at the the structure of the DNA under that SNP. And you can see that right down at the bottom, what that sequence is the consensus sequence for an ELF5 binding site, but STAT5 also binds in that region. And if you compare the wild type version of the SNP, you have either a C or a G there present, depending on which strand you look at, in comparison to the mutant, which is T or A. There are tools you can use that will predict what happens to the binding activity when you mutate the SNP. And so that's the prediction that's shown in the bar graph. Now, what Monique did in the lab after that was to use Gel Shift Assay to try and actually see, first of all, if we do this, do we see a difference in nuclear proteins binding to the site? So this is a gel shift where the labeled oligo is either the wild type version of the SNP or the mutant. So the C allele is the wild type. So you can see in the C allele, when we add the probe, there's a shift, and you can see that faint band up near the top. It's competed off when you add unlabeled oligo. And if you have an unlabeled mutant oligo, you can see it's not competed off. And of course, the other side with the T allele, there's no binding at all. So at least in the in vitro circumstance, this suggested that, wow, this might be worth looking at in terms of other aspects. It turns out that when we look at the expression of our genes, all right, this I put in there to just kind of give us a sort of a thing to think about. There's a diagram of mammary cell secretion from a review by Bo Lauderdale a few years back, showing where these potential things could work. For example, SLC40A1 is thought to regulate vesicular transport of iron, so that might be involved with secretion. And 9A4 and 9A2, these are not iron transporters, they're sodium proton antiporters. So they can potentially work by setting up a gradient, a proton gradient that then drives the energy for other transporters to work. So what we did then was to then just take mammary tissue from strains of one genotype or the other and compare the expression of these genes in the mammary gland. And that's shown in this real-time PCR analysis of the 4 transporter genes. So the black 6 strain in dark blue is the wild type, and the 129 is the mutant. And the bottom line is that if you compare across the 4 genes, there wasn't really essentially a big effect or no effect at all on the expression of these at the RNA level. So that if that STAT region is involved, it's not really having a big effect in the intact gland. We measured protein levels as well for some of these. And so this is immunohistochemistry, comparing 129 with black 6. And again, there weren't any differences there. So just summarizing what we got out of that paper, we had identified several loci. We had one that was particularly interested in. We're still somewhat interested in trying to follow that up, but we've had other things to do. There were 4 transporter genes, and the expression was not influenced by genotype. And this sort of identification of candidates and then trying to follow up on them using a slower method is kind of something that we've struggled with, but others have struggled with it in the GWAS field as well. And so there needs to be other approaches to try and really kind of connect this genetic effect with phenotypic effects. We just decided to change sort of our tact a little bit after this, and we had measured all of these traits and we had the data, so we decided it was really time to go in and just run the analysis, the GWAS analysis on everything that we had measured. So that's what this heat map shows here. You have the location in the genome on the bottom, and then the left side of the heat map are the p-values for the SNPs. So if you look at the code, the key, the dark ones and the dark blue are not significant. And then when you start to see cyan, green, and yellow, those are becoming highly significant. So what did we get out of this? Well, this sort of just is to remind me that we have these different classes of maternal traits that we're looking at. And we ended up, if we used the 10 to the minus 6th cutoff of about 6,000 significant SNPs over 1,000 loci or so. So we have a lot of work to do if we're going to try and understand these. And this is where we started thinking we needed to have more of a kind of a systems approach, a broader approach using other types of data, and that's where we've been going. But the other thing I want to point out here is that we had these limitations that we were still working with. Firstly, we had missing genotypes, and I'll talk about that a bit. Limited corroborating evidence, things like genome-wide RNA-seq in all of the strains. And then this idea of maybe we needed newer computational approaches to try and use for prioritizing which loci we wanted to pursue. So this was the state of the variant data at the time we got started. That was all that was present. There were about 100,000 variants that we knew of, published by the Broad Institute. And these next graphs are going to basically be the same. On the y-axis, you have variant count and the strains on the bottom. So then there was this big effort from Perligin, and by 2014, that had produced a data set of about 600,000. You can look over on the right, we still had an issue with our high lactation QSI5 strain because no one was really kind of interested in that enough to sequence it. So anytime we found something that we were interested in following up on QSI5, we were kind of almost out of luck because we were missing the data. Then the group in the UK at the Sanger Institute went back and resequenced a whole bunch of strains, Thomas Keen's group. And so if you look at the y-axis, you can see now we're looking at about 5 million variants across the different strains. And even with this, there were 9 strains within our cohort that hadn't been sequenced. And so we decided we needed to do that. So a year and a half ago, we submitted samples, and then Thomas Keen and his postdoc Anu helped us map the reads and then start to try and interpret this. So now our situation is quite abundant. You can see we have on average about 5 million variants that we can look at. The one in the middle sort of, I'm sure is jumping out at you. That's a strain called the Czech strain, which is a wild derived strain. It's very different from the rest because it's only been sort of bred in captivity for about 40 or 50 years. And so the other strains tend to not have as much variation because they're, whether or not you like it in your breeding animals, there's a selection process going on and their variation is much less. Okay, so a little bit of to talk about challenges with these variants. And so I present on the top, there's a diagram that talks about the different kinds of variants we're studying. Single nucleotide polymorphisms, indels, which are 3 to 6 nucleotides, and then structural variants. If you go down into the graph in B, this is something of interest to us, and that's what are they doing? And it depends on where they're located. So we have intergenic SNPs, which are outside the genes, and then we have regulatory or coding region SNPs, which you can find either within the regulatory sequences or in the actual coding region. And then, of course, on the other side is another way that we categorize these, and that's by frequency in the population. So on the x-axis, if you go from left to right, it's rare versus common. And on the y-axis is the effect size. And so it's generally known that in variants that are present in over 5% of the population, your effect sizes are generally pretty small, normally on the order of 10% or less. And then as you go towards the y-axis, the rare ones tend to have a larger effect. And something that's also been somewhat interesting over the last several years are this idea of very rare variants or private variants, which is what I'm going to spend the next several minutes on. But you know, these, in our particular case, are variants that show up only in a single strain. So if we have 51 strains that are now sequenced, and if we pull the variants from one strain and assume they're high quality and they're not present in any of the others, then that's considered a private variant and may have kind of a special case in terms of things that it's doing. So what to do with the data? Well, we identify private variants and the genes they impact. We're going to redo the GWAS because now, when we first did it, we had about 0.5% of the known variation, and now we have about 90%. And then integrate that GWAS work with the transcriptome, and then lastly, try and do some comparative work with the cow and the human. Okay, so this is the situation with our variants. In this case now, the green is a SNP, blue is an indel, and then the yellow, the orange are private variants, and of course the Czech is that one that jumps out. These are just the private variants. So check, even as private variants has about 1 million, and the rest of them have somewhere around 50,000 or so. And you can subtract, check out, and you see the sort of more accurate numbers of what's going on with those mice. So we filtered out all of these private variants, and then we started, first off, just categorizing them by what they do and where they are. There were about, there were a total of 1.7 million private variants that we pulled out. About 3/4 of them were in between genes, so that's the pie chart on the left. They're intergenic, but 1/4 were gene associated. In the middle one, you can see the functional category. 99% of them were non-coding, and this is not a surprise, and it is why a lot of people are interested in integrating variant data with RNA-seq data to study regulation of transcription or RNA stability and things like that. 1% were coding. And of course, on the very right was the impact of the coding variants. And most of them have a very low impact. There are 7% or so that are high impact, and then about 30% that are moderate. So then we can categorize these by what they do. So on the left are the high impact coding variants, and on the right are moderate and low impact coding variants. And the high impact ones generally result from things like frame shifts or introducing of stop codons or start codons or deletion of stop codons. The moderate to low impact ones, the biggest category is missense. And of course, a lot of the people in the human literature like to find these and then genotype them in cases and controls. You know, depending on what it is, if the amino acid change is severe enough, then maybe there will be an effect. In terms of the non-coding variants, the ones that were the most interesting to us was the group on the left, the modifiers. About half of those are what they called regulatory region variants. And so these were probably within about 5,000 base pairs upstream or downstream of the gene. And some of them potentially overlapped with known regulatory sites. We also, if you look, there's upstream variants in the green on the left that are about 17%, and downstream are about 17% as well. And then on the right, these are considered low impact, and generally they have something to do with introns. Okay, so what did we do to try and now filter down this list to something meaningful? So we pulled the private variants, and then what we did was we compared them in our list of strains. And so every trait that we measured, we can set up a rank that's divided into quintiles. And then we can take the top and the bottom quintiles for each trait and get the gene lists for those, and then subtract them so we have a high specific trait list. So for example, the top quintile of milk production, only those genes that are in the high ones, and the low, only in the low. And then from those, we went into this data set database called InnateDB and used that for pathway analysis. So about 70 or so traits that were plugged through this, and I have a technician in the lab who spent quite a bit of time just doing this. And then from that, we would then sort of organize them and sort them for subsequent analysis, where Monique would then take our candidates and line them up with other regions in the genome. So I really hate to present this data as tables, so I like this. This is a word cloud that basically shows the traits that we've measured. The size of the word cloud is an indication of how many pathways were significantly enriched for that trait, and then the colors are just different classes of traits. So you can see the largest one is in high RNA to DNA ratio. I need to indicate here we did remove the check from this analysis, because actually we've done it both ways, but the check, because it has 10 times more variance than everything else, it's a really high influence data point. And so we felt that anything we saw with that was really probably not going to reflect the reality of what we saw in all the inbred strains. And so, okay, so that the coloring is different categories. And so the orange ones that are large, they actually represent the milk fatty acids that we were able to measure. So again, there's high C24 percentage in the milk and high C10. Those were enriched for a number of pathways, as were a number of the other fatty acid traits that we measured. Now, what this is a summary of all of the pathways that we were able to measure. Sort of, I didn't want to show you massive numbers of word clouds. So what this shows you is basically the minimum false discovery rate, adjusted p-value, and that determines the size of the pathway. And then we've color-coded them by the frequency. How many of our traits did they show up in? So bright yellow was the most frequent, and we had 12 out of the traits that we measured that showed up for that, and dark blue was 1 or less. And so based on that, we have these top 5 that I initially talked about, but we enhanced the list to contain 7. You can see olfactory system is jumping out at you, and of course that was the most significant, but that region in the genome is a particularly scary region in terms of trying to understand it, because it's just hundreds and hundreds of olfactory receptor genes that are, I'm sure they play a function, but how one person can sort that out is somewhat of a challenge. And so we decided to stay away from that for now. But the pathways that we ended up looking at were pentose and glucuronate interconversion, signaling by notch, PI3 cascades, steroid hormone biosynthesis, drug metabolism, glycosaminoglycan metabolism, and diseases of glycosylation. And then lastly, there was a sort of a general transport category, transport of glucose and other sugars. There's a lot: bile salts, organic acids, metal ions, and amine compounds. And so the bottom line is, from these 7 pathways, now we've taken our 200,000 or so private variants and filtered that down to about 3,000 in about 229 genes. And this is when Monique takes over usually. So I want to just spend a minute and talk about this concept of super enhancers. This is a thing that started really to develop about 5, 6, 7 years ago. And there was this idea that in the genomes of cells that were terminally differentiating into specific classes of tissue, that you needed these clusters of transcription factor binding sites. They're usually about 12 kilobases in size. And one of the main common things is that in addition to these histone modifications, they contain transcription factors that might be sort of specific to stem cells, and then this one factor called MED1, which is thought to be a factor involved with opening chromatin. So the thing that really sort of is interesting about this now is that, and I was fortunate enough to get a slide put together for this from a Gordon conference that was about 2 years ago, but Lothar Henninghausen's lab has done a lot of work on a memory STAT5 super enhancer class of regions in the genome. And this is a paper from a review that his fellow published. But essentially, there are about 500 or so of these STAT5 binding super enhancer sites that are thought to play a major role in determining how the mammary gland becomes activated and those luminal secretory cells produce milk in high quantities. And so if you look down in the center, these are the binding sites that are present, as well as how the chromatin is changed. And there's an example of one of the first ones that he talked about in A up there. So there's a STAT5 binding site right in the middle of these 2 STAT5 genes. STAT5B is being transcribed in one direction and 5A in another. And there seems to be some kind of positive feed forward that really brings the expression of 5A up. So this was one of the things that we focused on when we did our overlapping, and that's shown here. Again, this is a similar approach where you, again, are looking for modifiers. So the table is really the important thing here. And it started with about 3,000 genes in 229, 3,000 variants in 229 genes. 14 of them were associated with super enhancers, and 10 of them overlap with binding motifs, and 5 had variants that affected these genes. And so we now have a list of 5 genes from just doing the pathway analysis. And so the pathways they were found in are shown above. These were also found to disrupt those transcription factor binding motifs shown there, and the genes are listed at the bottom. So there's a divalent metal ion transporter, and then there are two sialyl transferases, steroid alpha reductase, and then lastly, a gene involved in glycosaminoglycan degradation, the sulfatase at the bottom. So these are candidates that we've kind of at least got a reasonable reason to query more for function. The other approach that we took was to make use of some RNA-seq data that we had collected from tissue we had from high and low strains on day 1 of lactation. And that's this slide sort of summarizes that. So the graph on the top left shows a collection of 3 high and 3 low strains. What's shown here are just some tissue weights, but then at the right-hand side, you can compare the weights of the dam and the growth, the weight of the litter. The heat map on the right-hand side is just overall gene expression values to cluster the traits together so you can show how the gene expression relates within and across different traits. And by and large, they largely clustered together. And the most interesting part is the table at the bottom. So these are the differentially expressed genes. We had pairwise comparisons that are shown in the lower left-hand diagonal. And so the thing that we did here was then to basically say, okay, of those private variant affected genes, which ones of them are differentially expressed, high versus low? And so by doing that, if we say, okay, look, if we look at BTBR, which is a high gene, and compare that to all the low strains, there were 24 genes that were elevated in that particular strain. In contrast, if you look at AJ on the other side, there were 16 genes that were differentially expressed in that in comparison to the high ones. And so this gave us a list of 56 genes and reduced our list of variants again to about 700 or so. And when we overlapped this with the super enhancer, we ended up with one candidate. It was in the strain AJ, and this gene, which is a predicted gene, GM36677, is a long non-coding RNA. So we're not sure what this does yet. And in fact, this data is really new to me. So we're going to have to think about this a little bit. But it actually disrupts a zinc finger binding site. And you can see sort of a little bit about it on chromosome 15 below. Okay, so I'm almost done. I think I only have a few more slides. I guess it's a good thing. I want to just change directions a little bit and kind of talk about this idea of, we're kind of thinking about what we do with GWAS results in the wrong way. I mean, the main thing that really has come out of a lot of the work is that all of these loci we identify have small effects. And so sometimes it's kind of hard to imagine how a single association, even if it's highly significant, could be causing an effect in your trait. And that's really shown in this histogram up on the top left. You can see effect size on the x-axis and the density of SNPs, or the number of SNPs on the y. And you can see, again, that by and large, most of the SNPs have an effect size of less than about 10%. Now, this has caused a few people, and there's a group up at Dana-Farber Cancer Institute who have thought about this. Some papers published 2016 and 17 that because these SNPs act with small effects, there's a good chance that they could act in clusters or as groups or modules. And so several have started to look at SNPs in a network sense. And this one is really kind of particularly interesting in me because of the way they've decided to sort of score it. So this is on the bottom in B, this is gene expression data that was obtained from a resource known as GTEx. For those of you who may not be familiar with this, GTEx is this huge effort that was done in the human to do RNA-seq on 50 or so different tissues. And they have 700 individuals that they've been able to do this on. And that allows them to map something called an expression QTL, which is essentially you're using the gene expression as the trait, but then you still associate it with the variants. And so that's what's shown here is the organization of these expression QTLs and the genes they regulate into a network. And so each circle represents a community of highly connected nodes. And then in between those, you can see connections, and the connections are illustrated by lines. Okay, I should put this, but this really shows how the network is constructed. You have a SNP on one node and a gene on the other, and then those organize themselves using these algorithms that sort of, they maximize the modularity or the number of different communities that you can detect. So the interesting thing that this group did was once they were able to sort of organize this, they then looked at something called the connectivity of any one member of the network to the others. And that's shown in the graph at the top, and it's called degree. So that's the x-axis. And the higher the degree, the more connections there are between any one particular node and the others. So the y-axis is just the frequency of the variance. And if you go all the way over to the right, you have a connectivity of, or a degree of about 100. And those are not very abundant. And the people who've written about these probably consider those variants that are present within genes that are really, really important and would result in death of the organism or something like that, something that's a master regulator. And then as you go down in connectivity, you can see the frequency of the variance goes up. So the first thing they did was they showed that when you started to look at these lower degree variants, that you could see there was the presence of GWAS SNPs. In this case, they're looking at GWAS SNPs from a disease state, COPD, but those tend to show up in these lower degree SNPs. And they came up with this metric called the core score, which is shown down in the bottom. But what it says is that if you have a member in the network that is highly involved with its module, it's more likely to have an impact on a disease state than one that is connected across the entire network. So that's something we're trying to move towards in our own work. We're starting to collect data for our own eQTL analysis. And there's also this huge resource that's available that I would sort of love to tap into. But one of the things that really piqued my interest in terms of the cow is the work that this group from New Zealand has done. And this one paper published a few years ago, they have RNA-seq data from 357 different individual lactating cows. So they've done eQTL analysis, and every few years they release a paper on one little piece of it. We know they have probably a huge network somewhere back there that they're mining. And then they have whole genome sequence from 500 or so cows, and they have imputed variant calls for 12,000 genomes. So this is something that I think would be really useful to try and, first of all, organize it into a network and then start to compare it across these different species. And that's something I'm going to try and lobby for. And really the future direction here is to really integrate expression data, which is shown in those color-coded boxes from different species, analyze their importance using the networks, and then that should allow you to go back and, if they're species conserved, have something that may be more viable in terms of hypothesis testing for function. All right, so this is just my summary. I've used almost every bit of my time. So we've got an enormous opportunity now with this whole genome sequence that we have. We've been able to pull the private variants, and hopefully we've got some candidates in the low-hanging fruit. And the pathway enrichment has allowed us to kind of focus a little bit on 5 genes, and we're integrating this with transcriptome data, but it's clear to me that we need to get some other sort of species involved that are lactating, and the cow is the most obvious one, and then last, different analytical approaches are needed. All right, I'll just spend a minute and thank my collaborators. Peter Wilson and Jerry Way, University of Sydney. We started together on this very early. I couldn't have gotten any of the sequencing data done without the help of Thomas and his postdoc Anu, Monique, of course, and Yu Xing with the epigenetic marks, and then something that I hope to get more on in the future, work with Kevin Harbatine and Cesar on the fatty acid profiles in the milk from these animals. I'll stop there. I appreciate your attention. And I'll try and answer any questions. Thanks.